And based on these observations, so people try to classify acute myeloid leukemia. And when you go back to the 60s and 70s, what people did is they took blood or bone marrow from a patient and looked at it through the microscope. And based on the size, the shape, and also the color and the distribution, like the ratio between the nucleus and the cytoplasm of a cell, they defined different maturation stages, and which they summarized in the French-American-British classification, which was established in 1976. And M stands for morphology, so it's morphology 0 up to morphology 7. And these are all distinct classes, but what's really important, so first of all, these classes are usually, there's not a strong association with outcome. And the other thing is, it really depends on the people who looks into the microscope, who makes the actual diagnosis. So you have to be experienced to do that properly. And there were like experiments where they gave the same slide to different experts of the field, and then they got, like, you get 5 experts, you get 5 opinions. So this is sometimes a little bit harder to do, and it's not very objective. And that's what we actually need to classify leukemias. So a different way of looking into this is when genetics, meaning that we can actually look inside leukemic cells and see how the information is processed. Do they have genetic defects? Is their information the same as a healthy cell? Yes or no? Like, these things became more prevalent in the 60s, 70s, and 80s, and eventually the World Health Organization picked it up and formed a classification out of it. And when you compare the WHO classification from 2012, you can see that there's leukemia with a translocation 821. So this means, like, I will show a picture of that later. And this means that these chromosomes exchange parts of each other. And there are other translocations, but it's only like 5 different ones. When you look at 2016, this list is much longer now of the ones which have either mutations or translocations. So, and this will become even longer once we, with the next classification, which will come out most likely in 2020 or 2021. Sequencing approaches and showed that leukemia, and this is why this classification is so important to develop it even more, is that acute myeloid leukemia is not a single disease. It's a group of diseases. So it's kind of the same thing when I ask you, like, what kind of car do you drive? And you say, like, well, I drive a Mercedes, and that's my German answer. And then I wouldn't still know what kind of car it is. Is it a convertible? Is it an SUV? Does it have automatic transmission? Is it black? Is it silver? Is it white? Is it a fast car, a slow car? And this is exactly the same way how we look at leukemias. So we try to put them into individual doors. We really try to classify them to figure out what leukemia is growing in this particular patient. Unfortunately, our classification is ahead of treatment options, and you will see this later, because ideally we would like to have for each leukemia a specific treatment, and that's what we are aiming at, and that's what we're working on. But classifications are much easier to do than finding treatments for that. But you can see here in this pie chart that we have more than 15 different classifications and genetic subtypes of AML. And this is really complicated. But it also makes it necessary that we do proper diagnosis. One way of looking into this, because we learned that genetic abnormalities also determine the risk category. So this is a classification from the European Leukemia Net, and what they did, and this is different compared to the WHO classification, where they were just listed as different groups. Here, the authors tried, based on treatment outcomes, tried to classify leukemias into favorable, intermediate, and adverse risk groups. Basically, if you have, for example, if you carry an 821 translocation here, your outcome is better than having a, for example, a complex karyotype or an inversion 3. So this is really important for us, because we also have to stratify not only how intense we have to, like, that we have to treat patients more intense, or can we treat them less intense? And this is important to know. The most frequent group in here is the intermediate group, where the normal karyotype, meaning like these patients have no translocation. They have maybe molecular mutations, but there's no major translocation defining this group. This is the most frequent group with above like 50%. And here, this is a good picture showing that how these gene fusions